Okay, good morning. Good morning, ladies and gentlemen. I'm Jong from AffyXell Therapeutics. So we are developing a unique and proprietary cell and gene therapy, which is potent, fully allogenic, off the shelf, and reaching in an industrial scale in realistic and practical ways. Okay. AffyXell Therapeutics is a joint venture based in South Korea between Korean big pharmaceutical company, Daewon Pharmaceuticals, and British biotech AffyXell Life Sciences. Daewon focused on establishing a very unique allogenic cellular platform using pluripotent stem cell derived mesenchymal stromal cell, designated as DWMSC. Above all, Daewon could see the potential of DWMSC to be much more potent by genetic modification, and fortunately could find the right AffyXell's Affimer platform, a small but powerful non-IG scaffold protein. Yeah, through the convergence of these two mother technologies, our AffyXell platform was created. AffyXell Therapeutics is an independent company with full-time management and employees, and also self-funded by seasoned investor syndicates such as venture capitals, government funds, and strategic investors. AffyXell Therapeutics is addressing a wide range of immune disorders with sizable markets and significantly high medical unmet needs. Okay, now let's discuss AffyXell as the platform technology by starting with DWMSC. Conventional MSC therapy derived from the adult tissue have some limitation of short proliferative capacity, and it creates issues of donor variance, product inconsistency in both efficacy and CMC perspectives. The WMSC is the parental cell of AffyXell, can overcome all these confronting challenges and limitations. Derived from the pluripotent stem cell, it has much enhanced proliferative capacity, enabling multiple passages in cell culture process and adoption of cell banking and batch production system that provide consistent, reproducible, and scalable CMC capability. The WMSC utilize wonderful cryopreservation media formulation to make sure the product off the shelf and delivered to each clinical site via ultra-cold chain supply measurement. So most importantly, we could evolve the MSC from the cellular therapy to cell and gene therapy through the DWMSC. Yeah, AffyXell originated from the natural substances in the human body, so-called stapin A. It's non-IgG scaffold protein that can act as an antagonist or agonist by binding to a specific protein, a target protein like antibody molecules. Affimers are approximately 10 times smaller than antibodies and have a relatively simple structure without any post-translational modifications such as disulfide bond or glycosylations. It makes sure enabling the ease of production in both prokaryotic cell and mammalian cells without any affecting cell metabolism. Similar to antibodies, target-specific Affimer can be easily screened from the various phage display libraries with the 10 to the 10 Affimer diversity. Yeah, by combining these two comparable DWMSC and Affimer technologies, AffyXell as the next generation cell and gene therapy could have been created. AffyXell is an advanced stem cell-based cell and gene therapy, which has reinforced immune modulating therapeutic effect to treat autoimmune disease, transplantation rejection, and inflammatory disorders with the explicit mode of action. So we have currently 3 ongoing pipelines. Each of them has a unique and comparable manner for immune modulation. At the end of the day, they can be used for combination therapies as they share the same mother cells. Yeah, through these 3 pipelines, we will be able to find the optimal dose for different patients with different disease at different disease stages. So AffyXell platform can be used in 3 variations. The first, AffyXell can be designed to secrete soluble appemer proteins inside the patient body, and we call them in-body factory platform. Second, similar to chimeric antigen receptor, appemer can be displayed as chimeric appemer receptor on the cell surface of AffyXell. Lastly, exosome from AffyXell, so-called AffyXome, also display the appemer on the surface of exosomal membrane. Yeah, we can say it's another way to develop engineered exosome therapy. Okay, this slide is to present the development and manufacturing process of AffyXell platform. So from the pluripotent stem cells, DWMSC was differentiated and mother cell bank was generated in the GMP suite. By using lentiviral vectors, the genus of appemers were introduced to the mother DWMSC, making different AffyXells. We can design the appemers to be secreted and act as a soluble protein, or make the appemers anchored on cellular membrane, depending on the desired mechanism of action and the product concept. Transformed AffyXells were selected to be the master cell bank and working cell bank with GMP-compliant processes, and batch production can be done for consistent, reproducible, and scalable manufacturing. Yeah, cryopreservation formulation is also applied so that global logistics can be achieved. And at each clinical site around the world, the product will be sold and directly administered to the patient. Our AffyXell platform enables the true allogenic and off-the-shelf product. Yeah, it's critical to maintain inherited and intrinsic unique and excellent immune modulation of MSC, even after the genetic modification with the gene of appemer. To verify that, we compare the key characteristics of the MSC before and after the genetic modification. As you can see, as shown in this slide, the key immune modulation feature of TWMSC is well maintained even after lentivirus-mediated genetic modification in terms of cell surface markers, cytokine profiles, and immune regulator profiles before and after inflammatory conditions. Okay, let's look at AffyXell as the therapeutic cell, engineered to restore the immune balance and deliver the best, safe, and durable efficacy for patients in lots of immune disorders. Our lead program, AffX001, is the soluble anti-CD40 ligand appemer expressing MSC. And we are currently conducting IND-enabling preclinical studies. Ph.D. is a starting indication where the endpoint is clear and can reach the endpoint in a relatively short timeline. After having the clinical PhD data with Ph.D., we plan to expand to other autoimmune indications. We have additional 2 other programs with different targets and different structure of apemers to be expressed in the MSC. Yeah, and we believe we can address much wide range of immune disorders. Yeah, especially CD40 ligand signaling is known to play a pivotal role in activated T-cell mediated B-cell activation and differentiation, which is engaging in adaptive immune responses. As you can see in the left panel, both naive untransformed MSC and AffX001 have similar excellent function to modulate T-cell activity, and no significant variance in terms of the innate MSC originated function. What's the unique function of AffX001 is that it secretes anti-CD40 ligand affimers to block the CD40 and CD40 ligand interaction, which inhibit activated T-cell mediated B-cell activation. The red line in the right panel is the activated B-cell inhibitory function by AffX001 compared to the blue line of naive MSC, having no significant efficacy activity in terms of activated B-cell inhibition. Yeah, through this in vitro cell-based assay data, we could obtain the clue for gain of function of AffX001 over the naive MSC. We proceed to in vivo efficacy POC study using an NSG-based xenograft humanized GVHD mouse model. G1 is the normal control in the bottom line. G1 is the normal control. Neither disease induction nor treatment has been done. The red line G2 represents the GVHD vehicle. You can see that GVHD has been successfully induced, showing high and increased clinical score. So G3 green line represents a naive MSC that you can see some significant improvement in the clinical score, especially AffX001, the blue line G4. You can easily find the much more enhanced efficacy in both clinical score and histopathological images simultaneously. Yeah, even non-GLP studies, we also conducted a pilot in vivo safety. And from these studies, any significant safety issue was not observed. This slide shows our second AffX002 pipeline, which is designed to stimulate regulatory T-cell activation and proliferation. Affimer target of AffX002 is still confidential, but I can say that it's known to be the master regulator of regulatory T-cells. The target to agonistic affimer and the paracrine effect of MSC will synergistically activate the regulatory T-cells up to the optimized level for the best therapeutic efficacy to realize immune tolerance. Currently, we have selected the target to specific agonistic affimer candidates and transformed read cell lines for AffX002 Affy cells. So as you know well, mesenchymal stromal cell is well known to be one of the safest and the most potent cell sources for exosome or extracellular vesicle production. Many exosome companies are eager to secure an authentic exosome engineering technology. We have a pilot data that our approach to introduce affimer gene into the MSC can be one of the useful methods to produce engineered exosomes. Affygen, as the exosome derived from AffyXell, is going to be another great potential area as the standalone therapy. Yeah, it's crucial for AffyXell to build partnership to establish an ecosystem for development. AffyXell has several key value networks with strategic partners for each developmental stage, from bench to bed. Okay, this year is very exciting for us as we are transforming into a clinical stage cell engine company. Currently, we are preparing for IND submission and first in human trial for our lead program by first quarter of next year. We are also advancing our two other programs and keep working to develop Affygen to be patentable. We are also actively seeking for development collaborations, so please feel free to approach me for any of your interest. Okay, with that, I would appreciate you guys for your kind attention. Thank you so much.